Hi guys! Welcome back again to my YouTube channel! For today's video, we are about to discuss the microbial genetics. But before that, please don't forget to subscribe my YouTube channel and don't forget to click the notification bell below for more updates and information about microbiology. Let's get started!
I just need to clear my head.
Microbial genetics. Genetics is the science concerned with the cell characteristics and how they are passed from one generation to the next. Gene. It is the unit of heredity and it is a segment of DNA that carries in its nucleotide sequence information for specific biochemical and psychological properties.
There are two types of genetics, the phenotype and the genotype. Phenotype, or the heritable physical characteristics of the organisms, or the eye color in humans, resistance to antibiotics in bacteria. And the genotype, it means the information in the DNA that control the phenotype. There are compartments of microbial genetics in curriculum. It is the basic genetics, genetic variation and gene rearrangement, and the advanced genetics. The basic genetics are the molecules of genetics. The main molecules of genetics are called nucleic acid. All the genetic information are stored as a sequence of bases through nucleic acid, mainly in DNA and in RNA or some RNA viruses. DNA or deoxyribonucleic acid. It serves as organism's genetic material. It is divided into two functional units or genes. Most of DNA is double-stranded, the two strands held together by hydrogen bonds between adenine and thymine, or guanine and cytosine. It consists of non-identical complementary base sequences. The basic structure of DNA molecules is the nucleotide. It includes the phosphate group, pentose sugar, and nitrogenous base. First is the sugar. It is a cycle form of 2-deoxyribose sugar that forms the backbone of the DNA. Second is the phosphate group. It has a backbone and is the portion of the DNA double helix that provides structural supports to the molecule. DNA consists of two strands that wind around each other like a twisted ladder. Each strand has a backbone made of alternating sugar or deoxyribose and phosphate groups. Phosphate structure. The chemical structure of phosphate is PO4 to the power of -3, where a central phosphorus atom is bound to 4 oxygen atoms. One of the band is a double bond, and the other 3 oxygen atoms have simple bands, and over such oxygen a negative net are present. The third is nitrogenous bases. Cyclic structure of purine and pyrimidine rings. There are two major purines, the adenine and guanine. 3 major pyrimidines, the cytosine, uracil, and thymine. Sugar bonds are linked to each other by phosphodiester bonds, a single phosphate connected by ester linkage to 2 sugars. DNA consists of alternating units of phosphate and 2-deoxyribose. Phosphate connects to 2 sugars, bonding the 3 carbon of one sugar and the 5 carbon of the next sugar. DNA is only polymerized 5 to 3 and as antiparallel. One strand in the direction 5 to 3 and the other strand polymerized in the opposite direction. DNA structure. Prokaryotic DNA molecules are double helix and circular. These circular double-stranded DNA molecules are twisted and compacted through a process called supercoilings or superhelicity. This is done naturally by topoisomerase enzymes. RNA or ribonucleic acid. Structurally similar to DNA except most of RNAs are single-stranded. Second is the sugar is ribose instead of the oxyribose. And the third is uracil base instead of thymine base. Functionally different from DNA. Some of RNAs are used as messenger molecules or the mRNA to transfer information from DNA to protein. Some of RNA as a part of ribosomes. Some are adaptor molecules. And the last is few RNA only acts as a genetic material like DNA or the viruses. Example of RNA is the genome. It is the total genetic information in an organism. Prokaryotic genome or the bacterial consists of a single copy or the haploid circular DNA molecule. It ranges from 580 to 4,600 kbp. Many bacteria contain extrachromosomal DNA materials as a part of genome called plasmid and transposon. Eukaryotic genome or the some fungi. It carried on two or more linear chromosomes separated from the cytoplasm by nuclear membrane. Diploid eukaryotic cell contains two homologous copies of each chromosome. Eukaryotic cell contain mitochondria and chloroplast. Some eukaryotes like yeast contains plasmids. And the last, eukaryotic gene, unlike prokaryotic, are interrupted by intron. Viral genome. Unlike others, may be of DNA or RNA, capable of survival but no growth in absence of host cell. Replication of viral genomes depends on the metabolic energy and the synthetic machinery of the host cell. Most viral genomes range in size from 40 to 300 kbp. DNA functions in microbiology. First is the replication. Second is the gene expression or protein synthesis. It includes transcription, translation, and post-translational processes. The original DNA model by Watson and Crick. X-ray diffraction photo of a DNA molecule. DNA replication. Semi-conservative, which means that one DNA molecule gives two DNA molecules, each one consists of one strand from the original DNA and the other strand is newly formed one. Replication starts a fixed point called a region of replication and terminated at fixed point called TER or fixed point. Steps of replication. First, the two old strands of DNA are separated by the helicase enzyme to form the replication fork. Second is the replication of one strand template, and 5 to 3 direction can proceed in a continuous fashion starting by addition of short segment of small RNA at the starting point called primer. On this primer, DNA polymerase enzymes start to synthesize the new strand in a continuous manner from 5 to 3. This is called the leading strand. The third is the replication of the other template strand can only proceed discontinuously. This strand is called the lagging strand. Many primers or RNA primers are used to synthesize the lagging new strand as fragments called Okazaki fragments. The fourth is all of the RNA primers are removed by DNA polymerase 1 and the gaps are sealed by DNA ligase to make a continuous DNA strand. Fifth is after the formation of the two strands of DNA, the old one and the new one, DNA-gerase enzymes or topoisomerase II make a twist at the superhelicity of each DNA molecules. Remember the enzymes of replication. It includes the helicase enzyme, RNA polymerase, DNA polymerase, DNA ligase, and DNA-gerase or topoisomerase II. Sequence of events occurs as follows. Each gen along the DNA is bounded by promoter and termination sites. One promoter recognition, it is the area at DNA recognized by RNA polymerase before the gene to be transcribed. Two-chain initiation, in which recognition protein called sigma factor attaches to RNA polymerase to put it at the correct site of the first nucleotide to be transcribed. Three-chain elongation, in which RNA polymerase synthesizes the whole length of mRNA by polymerizing the complementary bases in the 5-3 direction. Note: RNA polymerase does not need primer to start, unlike DNA polymerase. Four-chain termination. RNA polymerase continues until a transcription termination signal or terminator where the polymerase and newly synthesized RNA dissociate from the DNA to end the transcription. That's all for the microbial genetics.
Like this video, please don't forget to like, comment, and share for more updates. Bye guys! See you on my next vlog.